Okay, so first of all, I would like to thank the organizer for inviting me to this exciting event. So my talk is, the title is Statistical Physics of Growing System, and this is really a way of somehow bundling together through a common theme a number of different projects that we have been thinking about in my lab. So let me introduce this topic. One very basic feature of biological systems is that they typically grow. So this is one of the oldest, perhaps, observation in biology, that if you have, in this case, a culture of yeast in the presence of a nutrient-rich medium, you would observe that the biomass will increase over time, typically in an exponential fashion for short times, and then in a saturated manner, reflecting the fact that resources start to be depleted. So these experiments have been done for probably 100 years by now. With modern techniques, we can start probing this kind of phenomena at the individual cell levels. And so we can start thinking about what happens to the cells inside these colonies and their, for example, their genealogic relations, and how, for example, properties at the single cell level, for example, duration of the cell cycle and their fluctuations, reflect into global population level properties such as the growth rate. And if you think about it, this problem really is connected and is fundamentally similar to the classic problem of statistical physics. So what we basically do in statistical physics, we have systems of a large number of identical units or individual molecular units. These units, when put together in a large number, they have some non-trivial collective behavior, and connecting between these individual dynamics and population level dynamics in a biological language is really the fundamental problem of statistical physics. Here there is a twist because of growth. This problem in growing system is very far from thermodynamic equilibrium. And so the idea here is this fact that these systems are out of equilibrium lead to new properties that you wouldn't observe in equilibrium physical systems. So I will not talk much in my talk about dimensionality, or at least not in an explicit way, but you can imagine that clearly this is a problem also that you can see as an instance of dimensional reduction, where you have a very large dimensional problem at the individual level, and you will reduce it to a few, let's say, relevant variables, for example, the growth rate at the population level. Okay, so this was, again, I wanted to just give you the general conceptual picture. And now I will talk about two specific cases where we analyze problems in which growth plays an important role. So the first is one of the main themes now in my lab, which is understanding dynamics of genome replications in growing populations. And the second has to do to the role of growth in pattern formations in tissues. Okay, so let me move immediately to the first part of my talk. So just to set the idea, we'll consider, and again, assuming that you are not necessarily familiar with genome replication, I will tell you about how this process works in the simplest case, which is, for example, E. coli, or pretty much 90% of the bacteria. So most bacteria have a single a genome that is organized in a single chromosome that is circular in shape. And when the replication starts, there are two large molecular machines called replisomes that bind at a specific site on the genome called the origin. And they start replicating it, one going clockwise and the other going counterclockwise. So this would be, let's say, an image at a certain stage of this process. And so you will see that there will be a template. There is the red line. And then there will be a newly synthesized genome that is the blue line. Everything shown in this picture is double strand, and the distinction between a template and the newly synthesized genome is arbitrary, because in reality, each one of them inherits one parental strand from the mother and a newly synthesized strand. So the idea that we had was to try to understand this process by looking at a set of experiments that biologists have been doing for a while. And this is an example of a paper that made me think about this idea. So what people do here is you grow a population of E. coli, in this case, in the exponential phase. You pick a sample and you sequence its genetic material using deep sequencing. And what you plot is how many fragments, when you sequence, you have to fragment DNA, the abundance or the fraction of fragments at a certain genomic position. So because the population is growing, you can see that this pattern is far from being homogeneous, and it presents a characteristic peak at the origin of replication and a minimum at the opposite site of the origin. And this reflects the fact that if the population is growing, you have many of these incomplete genomes. These incomplete genomes will always have the origin, and they will have a decreasing probability of including genomic locations far from the origin. And so what people have been doing is to use this pattern to see, for example, if you knock out some genes that are relevant for replication, how this pattern changes. But it's mostly has been used for qualitative analysis. And what we have been thinking is that given the improvement of sequencing techniques, perhaps we can use these kind of patterns to say something more quantitative about genomic replication programs. So I hope I don't have to specify for this audience, but of course, feel free to interrupt me at any time if you have any questions. So you're talking about this data?
Yeah, exactly.
Yeah, so we have a large collection of cells, and maybe I should specify already that these experiments are done on these cells are not synchronized, so they are at different stages in their cell cycle. That is crucial. It's possible to synchronize mostly. I mean, synchronized experiment is mostly done with eukaryotic cells. Synchronizing bacteria is extremely difficult. So this is actually will be the crucial point of the story. Thanks for the clarifying question. Okay, in fact, it was my next slide. So the problem is, if we zoom in this population, we would have cells at different stages in their cell cycle. And they will have a very different genomic content. So some of them will have a single genome, some of them will be just before division, and they will have 2 genomes. And depending, bacteria, to speed up the replication program, they have something called multi-round replication, which means in nutrient-rich conditions, they can start replication before the previous round has finished. So there can be also situation, again, this will be 2 different nutrient conditions where at the time of division, there are 2 genomes, 2 complete genomes, both undergoing replication. And there will be a mixture of all of this that will make up my population. So this becomes an extremely difficult problem to model. And the way we can get around this complexity is to ignore cells and just look at the genomes. And so the theory goes as follows. So what we assume is that we are in an exponential growth phase. So the number of cells grow exponentially at some rate. And we also assume that the population is in a steady growth, is growing also in a steady manner, which means that also the number of genomes as a function of time is growing exponential with the same exponential rate. And so from these 2 assumptions, I can say something about the age distribution of genome. So if I pick a random genome in my population, I can ask what is the probability that the process that has created it started time tau before the current time. And because you can think that the density of genomes of a certain age is proportional to the derivative of this function at a time t minus tau. Because the increase of the number of genomes gives me essentially the number of genomes that have been created at that time. And so if I evaluate the derivative of this function at time t minus tau, I will get something proportional to the age distribution of genomes. So from this, I can immediately conclude that the age of distribution of genomes is also exponential with the same rate. Yes?
Assuming that the population starts from one individual when you're doing the age distribution thing, because otherwise there might be a distribution from when you start, right? Other than just the derivative.
That's a good point. We don't start with one individual, but let's say the initial number of individuals is sufficiently small that I can neglect that effect.
Okay.
Thanks.
Thank you.
I mean, those will have completed replication anyway, so that would not really cause a bias. Other questions so far? So this is pretty much a classic classic argument in mathematical biology that is done for age structure model for example, distribution of ages of cells. But this is the same argument done at the levels of genome. Okay, so now I have to say something about how do I model DNA replication. And at this point, I don't want to be explicit. I will just say that there will be some model of this DNA replication program. And what this model will eventually produce is a probability that a certain genomic location has been copied at a certain time. So if the program is perfectly deterministic, this function can only be 0 or 1. So this would be an example of, say, a hypothetical bug that replicates its genome starting from 2 different origins, and the second origin is activated after a certain time delay. So then you will see here, the future light cone of the 2 origins. These are this black region. But I can also consider a stochastic case where, for example, origins are activated at random times. And in this case, this function will have some shades of gray, so values between 0 and 1. So from this function, I can also define the probability density that a certain location is copied exactly at time tau, and this is just the derivative of this function. So in other words, f is the cumulative distribution of psi.
At age 0, nothing is copied? What is it conditional on something? This probability?
At 0, nothing. So I define, let's say, the birth date of a genome, let's say when the first origin starts, or before the first origin starts. So
okay.
I can assume it by an appropriate choice of the origin of time.
Fine. Yeah.
Yes? Yeah, so what I'm saying is I haven't made the model explicit yet. What I'm saying is whatever model, simple or complicated, deterministic or stochastic, my model will produce a probability distribution, which is, let's say, maybe I can start from this one. So this is the probability density that a certain location on the genome has been copied at a certain time tau. And F is its cumulative distribution. So this is the probability that X has been copied before tau. So at tau, it is already present on the genome. And so this will be, again, if you have this origin and this origin, so at each origin, you have these 2 replisomes that travel in opposite directions. So this creates these light cones in this kind of physics language. And so this would be the meeting point of the 2 replisomes. That makes it clear? Yeah, okay. Yeah, this organism doesn't exist. This is just to say that this can also accommodate, for example, eukaryotes where you have multiple origins. Okay, so now we put everything together, and the idea is that if I want to know, so first of all, let me say, if I take a random genome in my population and I ask the probability that this genome contains a certain position, I can immediately express this probability as a convolution, because this will be the probability that this location has been already copied at time tau times the probability that the genome has age tau, and then I can integrate over tau. So this is just essentially Bayes' formula. I can also integrate this expression by parts, and I can write it in a more compact formula and say that the probability, again, the probability that this position is included in a random genome is the average of the exponential of e to the minus lambda tau over this distribution. Yes? No. So once 2 replisomes meet, they, yeah, that would be really, I mean, maybe it can be if there are some very serious genetic aberration, but in a normal situation, that doesn't happen. Okay. Okay, so again, this is pretty much the general theory that we will apply. And so the idea is that these experiments really measure this quantity, because if you have a large number of genomes, the proportion that you will sample in your sequencing experiment would be proportional to this function. And so now we have a very clear inference, Bayesian inference problem. So this is what we measure. We know the exponential growth rate because we have been growing these cells and we can measure how quickly they grow. And so what we want to do is to use this information and this information to infer this function and the underlying model that generates this function. So the first case we studied is wild type E. coli, so the simplest possible, the most commonly studied organism. So we performed these experiments at 5 different temperatures because temperature is a nice knob to turn for a physicist. It's a nice control parameter. You can see also the nice advantage of this technique is sequencing is improving really at an incredible pace. So now you can really generate high quality experimental data. So you can see that these curves are very clean. And you can see that there is clearly a dependence on temperature, which suggests that something in this process is being affected by temperature. Okay, so the first thing we tried is the simplest possible model that would, okay, so yeah, the first thing that we tested is, let's say these two machines go at a constant speed, there's no fluctuation, they start at the same time, they are perfectly deterministic. So in this case, this result is actually known for constant speed. You will get that this distribution has an exponential shape, whereas the exponential constant, and this comes to the question before, is given by the growth rate divided the velocity. Twice the velocity because both are copying. So this is nice because, as I said, lambda is measured independently, so you can use this as a simple way of determining the speed of replication. You can see that the fits are pretty good. And so with this method, we could see how the speed of replication depends on temperature. And you can see that there is about a factor 5 increase in the range we studied, which is pretty much reasonably well described by an Arrhenius law. I should also mention that there is also an Arrhenius law for the growth rate. So lambda also varies with an Arrhenius law, but the activation barriers are slightly different. And this essentially is why you will not see these curves collapse into each other. So the exponential constant is varying because lambda and v have 2 different dependence on temperature. So then what I was really interested in with this experiment is to measure the fluctuations in this process. So the idea is these are molecular machines, so they cannot go in a perfectly deterministic manner. The velocity must have some fluctuation, and that will determine some uncertainty on their meeting point. And so the idea is, okay, this curve has a cusp here and a cusp at the end of replication. And what I would imagine diffusion will do is to make this cusp a little bit more smooth. And so by measuring the smoothness of this curve, I can say something about stochastic fluctuations. So we have some information about this, but there is something else in this data that caught our attention. So you can see here that points are a little bit below the curve. Here they are slightly above the curve, they're slightly below the curve again, and so on. So we started observing this kind of small fluctuations, and to make it then more evident, I'm going to show you the ratio between the experimental data and this theoretical prediction. So at low temperature, if you do this ratio, you don't see much, but at all the higher temperature, you see this wave-like pattern. So here, the origin of replication is in the middle, so this is a asymmetric, like a mirror-like pattern. And you can see that it's present at all these 4 temperature, and this is a highly repeatable effect. So if you repeat the experiment independently, you will get the same pattern with the peaks in the same place. So to understand what it was, we first said, okay, let's assume that there are variations, like regular variations in the speed of replication along the genome. So we introduce a model in which the velocity is a constant times a sinusoidal term with a certain amplitude and a certain phase. And this model predicts these oscillations pretty well. I shouldn't say it predicts, this is really a phenomenological model. This is post facto. And now you can see that at the level of the abundance, what this model predicts is that this curve is really an exponential modulated by a sine. And so you can see, it's one of these things, once you see it, you cannot really unsee it. So you have this wave behavior. superimposed to the exponential. So this is, I think, is related with my previous slide. So when you, let's say, when you increase temperature, typically in the cell, everything goes faster. Let's say at least in a range where you're not stressing the cells. But so velocity increases and the cell cycle speeds up, but they do not go at the same pace. They're not proportional to each other. And you can think about it, this fact that they have to do this multi-round replication is because replication cannot catch up with the speed up of the cell cycle. So you cannot speed up the replication as much as the rest. Yeah. Yes?
My question is, how can you, in your model, is there a distinction between whether the oscillations arise from a velocity or from a growth rate? So if the growth rate itself was some not uniform process, but has some fluctuations in it, would that show up differently? Or is it just...
That's a very good question. So the only way we can do that is by looking at the growth curves. So we have the growth curves, and they seem pretty straight. There's nothing in the medium that suggests that there's something like that is happening. I mean, of course, it's a complex medium. These rich media are complex media, so there might be like very small effects. We also carried out longer experiments in a chemostat, and we found exactly the same result. So that suggests that you know, the medium is steady. So that shouldn't be the source.
I have a question about the oscillations in velocity here. So here you're assuming that the oscillations are independent across temperature, or you have basically one form that is the same across
So the model is a constant. The velocity is a constant times another constant times cosine omega x plus phase. And these parameters are fitted independently at each temperature. So if we do replicates at each temperature, we get pretty much similar parameters. I mean, there are slightly fluctuation because this is a small effect, but the parameters change a bit with temperature.
both the amplitude and the frequency change. Yeah.
Yeah, so I'll actually summarize it here. So this is pretty much, so this is the velocity predicted by the model divided the average velocity. So you only see this correction term if you want. So basically what the model predicts is that there are this variation with the, say, 15% of the mean. That's the typical amplitude. This doesn't change much across temperature. There's a slight variation in the phase, and there is this very long wavelength, about almost 1 million base pairs. And so we started wondering, what is this? Like, how do we understand this phenomenon? And we found, we didn't find really an explanation, but we found at least a corroborating evidence. And this comes from the fact that people have been studied in recent years variation of mutation rate in a bacterial genome across the genome. And what they found is that for many bacteria, the mutation rate is far from being homogeneous on the genome. And if you plot the case of E. coli on the same figure, this is what you find. So these points are experiments from a mutagenesis experiment. And this is just an interpolation using a sinusoidal curve. So you can see that these patterns are pretty highly correlated. And this is surprising because it's a completely independent experiment done from a different lab using different conditions. It's not even the same strain. So to do a mutagenesis experiment, you have to knock out the mismatch repair system. So you find the correlation coefficient are about 0.7, which is quite striking even. So this is very nice because it suggests something that also has been one of the themes of my research, which is the idea of speed accuracy or speed error trade-off. So the idea that maybe these two phenomena are linked together in a way that when in the regions of the genome where for some reason these machines are traveling faster, they're also making more errors and thereby causing a higher mutation rate. So I will not go much because this is a general talk about the possible cause of this phenomenon, but we have reasons to believe, do I have, oh yeah, I have a slide about this. So our hypothesis, current working hypothesis is that these oscillations are caused by competition among multiple replisomes. So I mentioned before that E. coli has this possibility of starting replication before the previous round has finished. And so our idea is if somehow replisomes can interfere with each other when the new round starts, the previous forks, the previous replisomes must have to slow down a bit because of shared resources, because resources are being depleted. And vice versa, when a round finishes, suddenly there is more resources, i.e. more nucleotide, and then forks can travel a bit faster. So there are two supporting evidence for this. One is these oscillations appear only in the temperature region where our model predicts that replication occurs in this multi-round fashion. And the second is that the period of the oscillations, if we look instead of as a function of x, as a function of time, seems to be proportional to the cell division time. So this is also the time scale of this seems to be consistent with this explanation. Okay, so the last thing I want to mention is that the theory I mentioned in the beginning is completely general, is not specific to bacteria. And so we got excited and started thinking about eukaryotes. So in eukaryotes, replication of genomes is more complex because what they can do is to start replication from multiple places on the genome. And so the function f that I defined before looked more like something like this. And so if you look at this picture with a background in statistical physics, you might think of a nucleation process, so like crystal growth, for example. And this analogy has been exploited in the literature, in particular in this very nice paper by the group of John Beckhofer, to model this genome replication program. So what you can do, and then this is again, this is a classic study by John Sommel, Avrami, and Kolmogorov. If you want to know whether a certain space-time point has been replicated, you can look at its past light cone. So all the points from which a replication program can reach this point by this time. And so this point, you can say that this point has not been replicated if no forks, no origins are fired within this light cone. And so what we can do is to use this approach to essentially factorize the problem and put it inside our convolution formula. So this is just to give you a sense of how the math goes for the eukaryotic case. We did a first attempt of using this kind of model to fit what happens in budding yeast. So we have this kind of model, and we let the origin of replication free, and we try to fit them, their location to the model and also their intensity. And so these are the results. So what you see here, so these are the data. The green are the data, the black is the fit of the model. These peaks are the origins of replication predicted by the model, their location along the various chromosomes of budding yeast. And the blue lines are the experimentally known locations of the origin. So you can see that there are some regions, for example, here, let's say here, where the model does extremely well. It really gets, is able to guess where the origins are. There are other regions that are more noisy, for example, this one. But these are, the data we used are pretty old, so now we are in the process of trying to generate more modern data with current techniques, current sequencing techniques, and hopefully we will be able to do better. So this concludes the first part of my talk. If you have any more questions. I think that would be very interesting to see. We are not, as you see, our precision for yeast is still not at that level. So I think at some point, maybe I'll ask you the reference, because yeah, when we manage to do like a better inference.
So how do you fit at the same time the velocity profile and the position of replication?
Well, I mean, this is Monte Carlo.
No, but what I mean is, I get you have like some likelihood landscape where you have multiple solutions, I mean, it's like seems an inference problem where you can have like multiple solutions that.
So, okay, so this integral, if you, let's say, fix the origins and fix the velocity, you can do it analytically. So you get a bunch of exponentials. And then what we did is like simulated annealing. But of course, we are not entirely sure that, this is not a local minimum.
But if I understood correctly, you are trying to infer at the same time the velocity oscillations and the position of the origins.
Yeah.
Right.
No, here there's no velocity oscillation. Here it's just.
Okay.
No. formula, because it would be too many parameters given the quality of data. So in this case, this is essentially, there will be the form for a single origin, and I'm assuming constant velocity, so this will be just okay. So as I was saying, the second part of my talk will be about something different, which is about patterns and growth. So I'll show you an experiment that we did after some modeling based on the idea that growth can lead to patterns that will be not necessarily possible in the absence of growth. So this is really thinking about, for example, developments whereby classic models such as Turing patterns do not really have growth. And growth is something that has been considered in more modern theory of development, but there are not so many mechanisms for pattern formation that fundamentally depend on growth. So this is a very simple experiment in which we have two species of neutral bacteria, E. coli, that are fluorescently tagged with two different fluorescent proteins, and they are reproducing and competing in a microchannel with two open ends. So in the beginning, they are mixed, and you can see they will push each other and they get expelled. And in the course of a few generations, you can see that starting from being randomly mixed, they create this stripe pattern. This pattern is metastable in the sense that it exists after a few generations, and after a while, you can see it start getting disrupted. So this is an experiment we did after some modeling that showed that this kind of pushing dynamics can create this unexpected stripe pattern. They're just a plasmid with two different fluorescent proteins. They are otherwise identical. Yeah. So I will not go into this detail. I just wanted to flash this because this is how we started to think about this idea. And what I really want to talk about is squids. So this is our most recent work on this idea. And we collaborated with a unit of some rhyteratoids who studies squids. So they're mostly interested in how their skin patterns are related to neural activity. But when I saw the work of some lab, what I was really curious about is this pattern itself. So even if you don't think about neural activity, cephalopods tend to have quite beautiful patterning of the skin. And the way this pattern is created is something that always fascinated me. So these black cells are called chromatophores, and they are used for camouflage. So what squids can do is control the size of these cells by neural signal. And this is done using some muscles that surround each cell. But again, you can also study this pattern regardless of the neural activity. And what you will do is you anesthetize these animals. And so if you do that, these cells remain of a fixed size. Although you can see that there is some variability in the cell, and this is due to growth over time. So cells are inserted in the mantle. They are small when they're inserted, and then they gradually grow with age. And so we started thinking, okay, how does this pattern look like? And what are the statistical properties, and how is this pattern maintained during the animal growth? So these animals can grow of several factors in size during their lifetime. So the first thing we realized, and this is in keeping with the literature, is that this is consistent with a mechanism in which insertion of new cells is possible only with a certain volume exclusion. So you can see that there is a certain minimum distance between the cells, and this is what we believe to be one of the main, and is also what is believed to be in the literature, one of the main forces shaping these kind of patterns. So one of the things because of this observation, we started thinking that perhaps we can understand this arrangement of cells using the theory of disordered system, in particular disordered random packing. And in this literature, there is one very prominent idea, which is hyperuniformity. And the concept of hyperuniformity has to do with the fluctuations of number of units you observe as a function of scale. So if you have, let's say, a generic point pattern, what you can do is to look at areas of different radius. Yes? It's a single cell. It's surrounded by muscles, but each black dot is a single. Yeah. Yeah. As far as I understand, yes. Okay. Yeah, so the idea of hyperuniformity is if you take sample areas of variable size, you can look how the mean and the variance of the number of points varies with this sample area. And so you can have, let's say, a simpler case in which you put points at random, so you have a Poisson point pattern. In that case, you would have said that the variance of the number of points would be proportional to the area. The average number of points would be also proportional to the area, and so you would have a linear behavior of the variance with the mean at changing size of the area. And so what people have been finding is that there is a broad class of dense disordered system for which you observe an exponent less than 1, which means that if you increase the sample area, the variance grows less than linearly with the size of the area you are sampling. And there is also a conjecture called the Torquato-Stillinger conjecture that says that all randomly jammed systems, maximally randomly jammed systems, are characterized by alpha less than 1, and so in this sense, they are hyperuniform. So there is this very broad class, this property that is satisfied by a very broad class of randomly jammed systems. So what we were thinking is this pattern is created by insertion of the cells at a minimum distance. This looks like a pretty dense arrangement, so perhaps this is a hyperuniform pattern. So what we did is taking pictures of this animal and looking at the scaling of the number of chromatophores we saw with the area. And this is what we found. So the green points are experimental data taken from snapshots of actual squids. And you can see that, so the line is a power law fit with exponent 1.663 about. So this is a straight line with exponent 1 for reference. So this is clearly quite larger than 1. So this goes in the opposite direction of what we expected. So in the literature, in the hyperuniformity literature, this is often called anti-hyperuniform behavior, or it's something that I cannot really pronounce, so we decided to call it hyperdisorder. And this was quite unexpected, because this is not something, so this kind of hyperuniform behavior has been observed before in some systems, in some randomly packed system, but it's a pretty exotic behavior. Yep. Is from different animals. Is the error of the variance, looking at variability across different animals. Yep. So we try to understand what's the physical mechanism that causes this. And by playing with different models, we found something quite surprising. And this is one of the nice cases when biology tells us something about physics. So we found that we could reproduce this behavior by simulating the simplest possible model you can imagine. That is a model of packed disks on a growing domain. So if I start putting a random disk on a domain with volume exclusion, so I reject insertion if the disk overlaps with a different one, then we found that this kind of dynamics has a very different behavior from conventional random packing. So let me run a simulation of this model. So now I'm in a rescale frame of reference. So instead of expanding the surface, you see the disks becoming smaller. And what you can see that after a while, you can start forming these regular domains. And this domain grow in a very intermittent fashion. And this is something you can see because if you nucleate a regular domain, you can not insert a new disk unless there is a space within each triangle where it's enough to insert a new one. And then as soon as that happens, then you can suddenly double the density. So the density grows in a sawtooth manner. And so you have this dynamical heterogeneity in your system where you have these regular domains of different size that grow in, whose density does this zigzag dynamics in an asynchronous manner. And this dynamical heterogeneity is what?
I don't know. Yeah, if the structure was not growing, what will you get as an exponent? You will get something below 1, right?
Yeah, you cannot get the larger than 1.
So basically, without the growing one, we will get something that is more uniform than random, I guess, because you have excluded volume, right? My question is, suppose that you have like this disk and you place this disk and they are not, I mean, you don't have growth.
I think if you don't have growth,
You have something that has an exponent below 1.
If you do without growth, you probably get 1, because to get below 1, you have to pack them. You really have to jump them together. I'm not sure if you can get type uniform by just placing them at random. This is actually something we tried. But for sure, you do not get larger than 1. We pick random coordinates and we try to insert disks, and we reject the move if the disk overlaps. That's it. And then we start growing, and the insertion rate, there's a separation of scale. So the insertion rate is extremely large compared to the rate at which we start.
You see a certain, almost like a lattice style pattern, even if the sizes are, changing, there is a...
I'll come to that in a couple of slides.
No. I'm saying, what if the way you're putting the disks, if you start with some structure in the disk instead of at random points, you have a certain rule that, okay, you would want to start at a given location, or rather around a given location, spaced out, and then that could also give you the same pattern. Maybe that's another possible way of it happening, right?
I'm not sure.
So
maybe can we postpone that to the end, because
sure,
I will share a few more things about this pattern, and then we can discuss whether you're convinced or not. Okay. So first of all, this dynamics, as I was anticipating, gives you the right exponent. So you get the simulation of this model. Now you can see a large scale snapshots, and you can see these domains of variable density. If you characterize them by their static parameter, you can also show a list from simulation that there seems to be a scale-free distribution of sizes of these domains. And as a consequence, what you observe is this system is hyper-disordered in that it has an exponent larger than 1, and the exponent is pretty much similar to what we observed in the squid. We have over many simulations.
And does it matter?
It shouldn't. You can get small, I mean, especially in experimental data, you can get some small effects because we are sampling a finite number of points. So we found it safer to average over squids. This is average over squids, and therefore.
And this 1.63 you just get for free by doing this process. Exactly.
Yeah. So I should say we don't know exactly. So we are taking this insertion rate to be very large. If we take it smaller, then the exponent start going down. So you go to a Poissonian phase. We don't know what happens if you increase it even further, because at some point this becomes computationally quite heavy. So we get it for free. We don't know, again, how, it's like in glassy dynamics, where at some point things become very slow, and yeah. Okay, so the other thing I wanted to mention is about the size of chromatophores. So what we assumed in this model is that the size of cells do not have any bearing on their exclusion zone, and therefore we could describe this point pattern without caring about the size of cells. But now we wanted to look at their size dynamics as well. And so what Robert did, the first author of this study, is he took pictures of the squids for about 1 month, from juvenile stage to adult stage, pretty much. So in this time span, they grow about a factor 2.5 in length. And he had this rig with multiple cameras to do 3D reconstruction. And so he could track chromatophores over time during this time period. So this experiment also confirmed that this seems really to be linear stretch. So it's pretty much similar to the model. But one observation that is interesting is if you look at the size distribution of the cells, this is pretty much stationary through this process. So these are the distribution of cell size. And so you are inserting the cells, and cells grow at the same time, but they do in such a way that they do not change, they don't really alter the distribution. So we did all sort of Kolmogorov-Smirnov test, and this confirms that these distributions are compatible with each other.
Distribution looks like very interesting. It looks like there is one characteristic size that is small, and then you can sort of. Do you have an explanation on that, or?
Well, we are kind of phenomenological. So essentially, we didn't really wanted to explain why they have this characteristic size, but we wanted to understand the stationarity. And what we did was really employing a theory which is very similar to what I showed you in the first project. So if you think about your stationary distribution, you can always write it as a convolution over thinking about the time when a cell was inserted. So you say, okay, each cell has been inserted at a certain time in the past, and there is a probability that it would have a certain radius given its insertion time and the age of the squid. And then there will be an age distribution that you can derive in the same way, but this is a different form because now we are not, this is not an exponentially growing process. This is something where the number of units grows quadratically with time because it's a surface. And so what we found is that if you, instead of having an exponential growth, you have a quadratic growth, this changes the rule of the game completely. And in particular, for this convolution, you cannot have a steady radius distribution unless this dynamics explicitly depends on the absolute time. So this means that if you add a cell when the squid is young, or you add a cell when the squid is older, these two cells cannot grow in the same manner, otherwise the pattern will not be stationary. And this is something we see in the data. So these are growth curves for each chromatophore, so their size as a function of time. And I'm ordering them. So this chromatophore was inserted after 2 days since the experiment started. This was inserted after 4 days since the experiment started, and so on. So there is a hierarchy. So the chromatophores that were on the skin for the same amount of time are larger if they were inserted earlier in the lifetime of the squid. And there is also this theory suggested a data collapse. So if you plot these curves as a function of the squid age divided this squid age divided minus the chromatophore age, this theory tells you that this should collapse into a universal curve, and this is what we observe in the data. So now we have this master curve, and we can generate now, somehow put these two sides of the model together and look what happens. So essentially now we have this volume exclusion model in which we superimpose the chromatophore radii, and this generates patterns that are pretty similar to what we observed on the squid. I'm sorry, these two figures are not on the same scale, but you get the idea.
The squid, it does seem like in the center there's a clustering of larger cells. Is there some, I mean, maybe that's just one sample, but.
This one?
Yeah.
I think it's one sample, and it might also be doing, so there is also another story. So there are many cephalopods. We picked this one because it's very transparent, and it has, these chromatophores are on a single layer of skin, so it's very convenient. But the downside of this animal is that it has a quite high curvature.
I see.
So that's why we had to construct this three-dimensional. You cannot flatten it out, so that might cause some visual. Yeah. So my time is almost finished. I just reached my conclusion. So I showed you that growth can lead to these interesting emerging. I just go to my conclusion, then you ask me. Population level properties. We can use these properties sometimes to do the inference problem. So looking at these large scale properties and use them to say something at the level of individual cells. And also in this last part of my talk, I show that growth can lead to surprising new physics in spatially extended systems. And I'd like to thank my group for doing all the work. In particular, Deepak started this replication business, and now he's a PI at Bauer Institute here in India. Florian took over this project on DNA replication, and Robert is the main driving force for the squid project. my collaborators, funding source.
Can it happen that the smaller blobs who are in the physical proximity can come together, or bigger blobs can divide in?
No, so this is maybe something I should have mentioned. So another nice thing of this system is because these cells are surrounded by muscles, there is no cell migration. So these cells have to stay in place. I mean, of course, the tissue grows, but they cannot move around the tissue. And that's also nice for tracking because also besides being the constituents of this pattern, they can also serve as a reference point to understand how the surface is growing. But yeah, they cannot merge and they cannot divide.
So even though the surface is growing, the blobs are independently increasing in size, but not.
They're increasing in size, but they cannot divide.
They cannot divide. Another question is there any relation with the number and the size? Like how many numbers you see, and what is the size of their blobs? Is there any correlation between them?
The size distribution is stationary and the number grows. So it's pretty much linear growth, which means that the number grows quadratically with time, as far as we can tell.
Yeah, but does it happen that if you have the average of these two is like there are 5 big circles and 10 small. So does it happen that is there any correlation between them?
No, because the size distribution is steady, so the average size does not really vary over time.
Are you working now? Okay. So no, the question I was trying to ask was, they may not change over time, but is there a spatial correlation that will happen for the size? Like, let's say there's a, like as even he was talking about, it was the bigger ones tend to have a cluster around the center, and then you have in the one on the left, and then that's not quite the same case in the ones on the right. So there's more bigger blobs towards the center than there are on the sides. Is that something?
So I think this comparison, again, is not on scale, so please not take it. But I showed it because, at least qualitatively, so these two mechanisms physically are independent, but when they put them together, you will get this fact that, so because you add these cells in the center of the triangle, naturally, this will give you this hierarchy whereby larger cells are typically surrounded by an hexagonal layer of smaller cells. Right? So this is what you see also here. And there are, of course, some defects because it's a random process.
Thanks.
Other questions?
Another question on the distribution of, so you characterize the sort of non-randomness with this relation between the variance and the mean. Have you tried other properties like the distribution of the ratio of second and first nearest neighbor? I mean, there are other properties you can, because like my question is that looking at the picture, it doesn't look like more cluster than random. It actually look over this. So if I had to guess the exponent from the picture, I will guess something lower than 1, right?
But this is also, I agree with you. So one argument, one way I convinced myself that this makes sense, one is I redo the simulation because I didn't believe it. But apart from this, this is not saying that it's more disordered than random. It's about how the disorder grows. So it's a statement about how the fluctuations grow with scale, not about how much fluctuation in an absolute sense you have. Coming to the other question, I mean, this is quite typical. Maybe I can show an extra slide. So we did all these things about pair correlations, and these are similar, but they are not really much informative. So maybe I can show this one. This is a, why didn't, maybe I cannot play because it's a, so if you have this kind of patterns, you should have an integral of the correlation function that diverges and a divergence of the structure factor in the origin. But this is something that typically you will never see in data. So you need so much statistics to see this. So what I can show you here is, so the correlation function, this is for the red is the experimental data, the blue is a simulation of the same size. And this is a R to the minus 2 behavior, which suggests that this diverges logarithmically in 2D. And this is a much larger simulation of the model. So you can see that this is a bit more clear. But you know, this is pretty much as we can do. And this is the structure factor where you have a similar behavior. Again, you see this small peak at the origin, but this is the same for hyperuniform system. Typically, these other statistics are even less informative. Like the sampling with the area is where you see things more cleanly.